speech.
This is a video about speech, so I'm going to ask you to stop talking and listen. Have you ever tried to talk to your dog? I'm sure you have, and I'm sure that sometimes they even listen. I'm also sure, though, that you wish they would talk back, and I'm sure that they would if they could. But they can't. It's tragic. Why are we as humans so alone in the world, talking to ourselves forevermore? How did we start talking? Why did we start talking? Are we actually the only animals that talk? And what is Fox P2? Could it be the long-anticipated sequel to The Fox? I'm going to talk about all of that. Stay tuned. Let me tell you a story. Once upon a time, in the faraway land of Great Britain, there lived a family afflicted with a mysterious illness. One of the grandparents, 4 of the parents, and 10 of the 24 grandchildren weren't able to speak properly. Remarkably, they presented no other noticeable morphological or mental abnormalities. They just simply couldn't form or understand words as well as the average person. Where do you start with such a conundrum? Well, let's see what this pattern looks like on a pedigree. This clearly indicates a dominant autosomal disorder. This family, called the KE family, ended up being humanity's first clue into the genetic origins of speech, the FOXP2 gene. The FOXP2 gene, found on chromosome 7q31, codes for a protein that acts as a transcription factor, which has a structure called a forkhead that allows it to bind to specific DNA sequences. This DNA binding domain was altered in the KE family by an amino acid substitution resulting from a missense point mutation, changing its shape and thus affecting its regulatory ability. Okay, so it's a transcription factor, but what does it do? Immunoassay studies have shown that FOXP2 is widely expressed throughout the brain and is especially prevalent in developing fetal gray matter. It has generally been associated with regulation of hundreds of genes involving central nervous system patterning, development, and function, including signal transduction, neurite outgrowth and axon guidance, neurotransmission, and synaptic plasticity. In other words, it is integral to the formation and regulation of neural pathways. It has specifically been associated with the CNTNAP2 gene, henceforth called the catnap2 gene, a neuroxin. The neuroxin family is a superfamily of transmembrane proteins found in neurons. Catnap2, or contact and associated protein like 2, specifically has been implicated in neuronal recognition, cell adhesion, and maintenance of potassium channels. The reason for this gene being of particular interest is that a homozygous mutation in it, leading to a non-functional protein, was found in a family with a history of autism and language regression. FOXP2 negatively regulates catnap2, and the two are believed to be involved in the formation of neural pathways that allow humans to develop speech. Interestingly, the KE family with the FOXP2 mutation and individuals with catnap2 variants both showed similar difficulty in reading and pronouncing nonsense words like woogalamic or perplisteronk. I have the wild-type allele for both of those, which is why I was able to pronounce those just now, but this would not be so easy for an affected member of the KE family. Take a look at these two structures, the inferior frontal cortex and the basal ganglia. Both of these structures are known to be involved in human speech development, and FOXP2 expression was identified in the fetal precursors to these structures. How do we test this theory, that FOXP2 is integral to the formation of neural circuitry necessary for speech? You may know that human experimentation is illegal, something about ethics, but could we use other animals we've deemed not advanced enough to care? And this is where I must come clean. Until now, I've made the FOXP2 gene out to be some sort of miracle gene that is only found in humans, but it isn't. In fact, it's possessed by every other vertebrate. It doesn't even only operate in the brain, it's also important for lung development. I know. That sounds a lot less sensational and fun, but I promise I'm not wasting your time. I will go more into the evolution of FOXP2 later in this video, but I bring it up now because it's important you all understand that other animals have FOXP2 as well. That is where the lab mice come in. To investigate the effect of FOXP2 mutation and vocalization, researchers replaced the mouse FOXP2 gene with a human one. They created 3 groups: homozygous mutants, which had the mutant human allele like the one in the KE family, heterozygous mutants, which had 1 mutant human allele and 1 wild-type human allele, and homozygous wild types, which had 2 wild-type human alleles. So technically, all of them are mutants, but the mice homozygous for the wild-type human allele will be referred to henceforth as homozygous wild types. Interestingly, the homozygous mutants were extremely developmentally delayed and died relatively shortly after birth because of their underdeveloped respiratory system. This aligns with the fact that no human homozygous mutant has ever been observed, since it is likely survival with two mutant FOXP2 alleles is impossible. The heterozygotes, however, developed normally and displayed similar cognitive ability as the homozygous wild types, much like the KE family. Differences were instead observed in the learning of fine motor skills. Heterozygous mice learned how to run on a stationary wheel significantly more slowly than the homozygous wild-type mice. Electrophysiological examination of known FOXP2 expression sites in the mice's brain revealed abnormalities in neuroplasticity, most notably in the dorsolateral striatum. In simple terms, the mice possessing the mutant human allele did not develop new neural circuitry as a response to learning in the same way that the wild-type mice did. Strikingly, humans with the FOXP2 mutation exhibit similar abnormalities in the striatum when executing speech-related tasks. Let's zoom out a little bit and think more basically. What about the anatomical structures that allow us to form words in the first place? After all, there have been instances of great apes being taught sign language, but even our closest relatives in the animal kingdom cannot mimic human speech because of anatomical differences. Sure enough, FOXP2 has been shown to heavily influence craniofacial morphogenesis, and thus may have significant effects on vocal anatomical geometry. Once again, we must turn to our poor friends the lab mice for answers. And we will again look at the homozygous mutants, or the ones with no wild-type FOXP2 genes at all. Firstly, it must be understood that the ability to make sounds in any animal lies in the configuration of the larynx and the vocal tract. Humans, for example, have a larynx that is more descended than our great ape relatives, and mice with mutant FOXP2 and cartilage cell types decreased laryngeal and tracheal cartilage, as well as an abnormal shape in the laryngeal thyroid, was observed compared to the wild-type littermates. This resulted in weaker vocalizations in affected mice, as well as abnormal frequency and duration. In a third of mutant mice, no detectable call could even be recorded. As you may remember from earlier, the homozygous mutant mice were plagued with multiple deficits that may have confounded these findings, but the changes in laryngeal and cranial morphology were marked. Obviously, giving mice two functional human FOXP2 genes did not suddenly give them the ability to speak. The conclusion we can draw from these experiments is that FOXP2 is heavily involved in the regulation of both neural pathway formation involved in motor skill learning required for speech, and the development of structures like the larynx that allow us to modulate our sounds to create what we call speech. Its involvement in skull reshaping processes also lend to implicating FOXP2 heavily in hominid evolution. Okay, so we've established some links that FOXP2 has to speech using other animals. But if all vertebrates possess the FOXP2 gene, what makes the human FOXP2 gene different? To figure that out, let's use mice as an outgroup. The human FOXP2 gene differs by only 3 amino acids compared to the gene in mice, which for the roughly 130 million years of evolution since the human-mouse split, places the FOXP2 gene among some of the highest conserved genes. Now, before we throw the whole theory away, let's look closer. When we do, we see that 2 of those 3 changes occurred after our split with chimpanzees only 4 to 6 million years ago, and both in exon 7. When this sequence in multiple individuals from across the globe were sampled, researchers found no amino acid polymorphism, suggesting that this variant is fixed in humans. 2 amino acid replacements and fixation in all humans in 5 million years is unusual unless we consider outside factors like positive selection. To prove this, researchers computed the ratio of non-synonymous to synonymous mutations. Think about it. DNA in a particular area of the genome tends to mutate randomly at a relatively constant rate. Most of these changes will be synonymous, meaning that the mutation does not affect the resulting amino acid sequence or any other function of the gene. Some of them, however, will be non-synonymous, and these will alter the function of the gene in some way. If those non-synonymous mutations turn out to be beneficial, they will be positively selected for and be more likely to become fixed in the population. This is why when we see a higher than expected ratio of non-synonymous mutations to synonymous ones in a gene, this is evidence of positive selection taking place. The result of this study, humans showed a significant increase in this ratio, whereas other lineages did not. In addition, they also looked for signs of a selective sweep. A selective sweep is defined as the so-called hitchhiking of alleles around the suspected beneficial allele, increasing their frequency by proxy. Now, obviously, FOXP2 is a very ancient gene, so to be clear, I am not talking about selection of FOXP2, but rather the selection for the modern human haplotype. The first thing they looked for is the frequency of expected low-frequency alleles. These would be fairly recent alleles that, under a neutral model with a constant population, would be relatively low in frequency. Sure enough, researchers discovered an extremely high concentration of low-frequency alleles around the human FOXP2 gene.
at some point in the past we had some common ancestor that couldn't talk, and then 2 amino acid changes in the FOXP2 gene, and we're ordering at drive-thrus and arguing about whether the Star Wars movies are good or not. I don't know how you got here, but that's kind of a tough question. But humans are the only ones that can talk, right? There's us, and then there's chimps. So speech must have evolved suddenly.
What my fraternal twin is describing is the de novo model of evolution. This model postulates that speech, and subsequently language, emerged rapidly thanks to some drastic change, or perhaps just the right change. So did it emerge suddenly, or did it evolve more gradually? Well, we aren't really sure. It would be great if someone invented a time machine and we could just go back and ask each hominid until one of them doesn't give us an answer, but until then, we have to get creative. As one of Darwin's students, Thomas Huxley, put it, believing, as I do, that the possession of articulate speech is the grand distinctive character of man, I find it very easy to comprehend that some inconspicuous structural difference may have been the primary cause of the immeasurable and practically infinite divergence of the human form from the simian strips. In other words, it is hard to believe that speech, which is only observed to this great extent in humans, evolved slowly, rather than something like FOXP2 being the straw that broke the camel's back, or mutation that made the chimp talk. Anyway, I'm going to talk about how speech may have evolved slowly. This is called gradualism. Under this assumption that speech evolved more slowly based on an adaptive behavioral response, researchers broke human speech down into 3 key components: audiovisuality, rhythmicity, and coordination during conversation. Because these behaviors cannot be determined genetically, nor can they be directly inferred from fossil evidence, in this section we will mainly be looking to our fellow apes and monkeys for signs of evolution. Combining sounds and movements in communication is not a trait unique to humans. It has been observed in frogs and spiders as well. Wait, spiders? What the heck does that look like?
Let's give a bit of encouragement. No, that's definitely a threat.
Oh. Anyway, I digress. Many animals have audiovisual communication systems, but primates and monkeys in particular show a trend of an increasing number of muscles and innervation of the face, suggesting that facial expressions have been selected for in our order. This makes sense since our faces deform naturally when producing certain sounds already. But what advantage does this generate? Evidence for any behavioral advantage is still tentative, but both humans and macaque monkeys show significantly better performances when following audiovisual instructions compared to unisensory inputs. This could constitute a basis for a potential behavioral advantage, greater comprehension. While primates do indeed share a similar relationship between audio communication and visual input, another component of human speech thought to be important is the coordination of rhythm between our utterances and our facial expressions. Human speech, when broken down to the syllabic rate, falls in the 3 to 8 Hz range. Maybe that's easier to visualize if I told you that's the same as 180 beats per minute to 480 beats per minute. If you, the viewer, happen to be a macaque biologist, you might be in the comments already saying, but Milo, macaque vocalizations fall in that same 3 to 8 Hz range. And you would be right, but the key difference here is that human facial expression during speech shows the same rhythm. This is not observed in macaques. For them, it's more open the mouth, make the sound, then close the mouth. Indeed, we are so obsessed with rhythm that studies have shown that disrupting this 3 to 8 Hz Goldilocks range in either the acoustic or the movements of the face results in significantly worse intelligibility. If we are committing to this idea of slow evolution, then where does this come from? Lip smacking could be our answer. Lip smacking is a behavior that has been observed in chimpanzees, marmoset monkeys, and nearly every other Old World monkey. It has mainly been observed during grooming interactions, and in some cases, face-to-face interactions as well. This is thought to be a potential origin of speech since it falls squarely in that 3 to 8 Hz range, making it distinct from other orofacial behaviors such as chewing and teeth grinding. As you may recall, the 3 to 8 Hz range is rather large, so this is not enough evidence to make lip smacking a compelling candidate. Another supporting bit of information, though, was obtained via X-ray cineradiography, where it was discovered that, whereas in chewing, the hyoid, lips, and tongue are highly coordinated, in lip smacking, a distinct uncoupling of the hyoid and other structures was observed. This is also a characteristic of human speech. Here's another thing worth noting. When human babies first begin to vocalize, we call that babbling. It isn't really talking, and this is reflected in an average frequency lower than that 3 to 8 Hz range, as well as being highly variable. Chewing, on the other hand, babies just kind of come out of the womb instinctively knowing how to do that. Now let's look at macaques. Similarly, baby monkeys innately know how to chew properly, and its frequency remains more or less unchanged throughout the life course, but they don't know how to lip smack like the adults. Like human babies, it starts out slower and more variable. They have to learn how to produce that speech-like rhythm, just like humans. And when it comes to human babies, I'm not talking about forming intelligible words. I'm talking about simply producing sound that sounds like words. This similarity in development suggests that the mechanisms involved in lip smacking may be synonymous with those in speech. This begs the question, is it realistic to think that lip smacking and vocalizations could have been united to eventually form speech in the time since humans diverged from chimpanzees? A prudent reference are our relatives the gelada baboons, who produce wobble vocalizations, whereby lip smacking is coupled with sound to create, well, a wobble. Their closest relatives, the yellow baboons, do not exhibit a similar facial acoustic behavior. Chimps and humans diverged at least 6 million years ago. Gelada and yellow baboons only diverged around 4 million years ago. So, there is a precedent for this kind of evolution in a relatively short span of time. The last key to human speech does not necessarily lie in the production of speech itself, as it has with virtually everything else discussed in this video. It is something that will surprise you to think about, yet it seems so obvious. Cooperation. We do not speak in a vacuum, of course. We need others to hear us for speech to be effective, or what's the point? This is possible thanks to vocal turn-taking. We take the natural flow of conversation for granted, but it's an ability much rarer in the animal kingdom than you might think. Naked mole rats, crows, dolphins, and even macaques participate in call and response vocalizations, but this is not the same as an extended and coordinated vocal interaction. Certain animals like songbirds and some monkeys duet, where there is some coordination present between individuals, but it is limited to sexual competition and territorial scenarios. And so, I will address a question I posed at the very beginning of this video. Are we alone? In a way, no. One other animal displays the similar cooperative cadence in communication: marmoset monkeys. Marmosets have been observed exchanging phi calls, a high-pitched vocalization that conveys information about their gender, troop, and even identity. Notably, these exchanges can last up to half an hour. It makes you think, what does a simple monkey have going on to talk about for that long? You may notice the similarity in rhythm of this exchange. The most unique aspect of these conversations is that marmosets will have them unprompted with conspecifics, that is to say, other monkeys that they are not currently or seeking to be pair bonded with, making this distinct from duetting. Since no other Old World monkey displays such a behavior, it is believed that this cooperative communication is a case of convergent evolution between humans and marmosets. One theory is that this may have provided an advantage in cooperative breeding strategy as prosocial behaviors became more prevalent. Prosociality, of course, being a very distinguishing feature of human culture in general. So, which one was it? Could it have been the amino acid changes in FOXP2 that occurred after our divergence from chimpanzees that caused this drastic change that allowed us to develop speech? Or could we perhaps embrace the gradualist model, where behaviors like lip smacking and duetting may have been precursors to what we call speech today? As with most things, it was probably neither, and both. Perhaps they worked in tandem. It's at times like these when I'm in awe of the complexity of this game we play called evolution, and the behaviors and abilities we develop that we take for granted. So next time you have an idea, say something. The next time you witness injustice, speak up. And the next time your mom tells you to take the garbage out, listen. Thank you for listening to me talk, and thank you for watching.